Okay. So this is exploring evolution in bioinformatics. This one should be pretty fast, though I guess I'm not going to get 3 chapters in one day. Damn. Yep, not even close. Okay, so anyways, exploring evolution. We have a little evolution thing. Cave painting, which... Structural comparison permits the establishment of evolutionary relationships. The similarity of a 3-dimensional structure can suggest evolutionary relatedness. 3-dimensional structures have determined for only a small portion of proteins. Gene sequences and amino acid sequences are more known because the 3-dimensional structure just takes so much longer to do, as we talked about in chapter 4 and such, right? The sequence database can be probed for matches to new sequences, and this is the main way the evolutionary relationships are done nowadays. The homologs are going to be descended from a common ancestor and can be detected by sequence alignments. By the way, this is such a superior method to the old method, which was looking at the structures of like tissues and bones, right? For example, do you know how many different times crabification has happened that they've detected now? 11 separate times organisms have evolved to become crabs. 11. What's the superior organism on the planet? Apparently crabs. What is the end goal of everything? Apparently crabs. What the fuck? Go talk about Occam razor, right? The most simple explanation is the right one. It evolves 11 times. That's not simple. Homologs. These are going to be 2 molecules that are derived from a common ancestor. Orthologs. These are going to be homologs that are present within different species and have similar functions. Like crabs, right? Shell. Can't eat me if I have a shell, right? All right. Paralogs. These are going to be homologs that are present within one species and often differ in their functions. Okay, so right here we see the cow. This is the bovine ribonuclease. It's a digestive or enzyme. And here we see the same sort of thing that's showing up in human beings, right? And so these are orthologs right here. We have the human ribonuclease, which is our digestive enzyme, right? Okay. And so notice they are actually not alike, are they? Yeah, there's actually quite a bit of difference between them. But if you look closer, you can start seeing some similarities, don't you? Why do they have similarities? They are ribonucleases, okay? No matter who develops a hand, they all have to kind of look alike, right? Why does a robot hand look like a human hand? Has to grip stuff. Yeah. Paralogs. So here we have the human ribonuclease. Here's the human angiogenin. This actually stimulates blood vessel growth. You can see they're actually surprisingly similar looking, and it's because they're paralogs. Okay. Okay, so the statistical analysis of sequence alignments can detect homology. The significant sequence similarities imply 2 molecules have the same evolutionary origin and similar structures, functions, and mechanisms. This can compare nucleic acid and protein sequences. So protein sequences are typically more effective because there's a lower likelihood of random agreement than the nucleic acids. The sequence alignments. So sequence alignments are going to be a process of systematically aligning 2 sequences with respect to each other to identify regions of significant overlap. And so right here we see this process right here of human hemoglobin, our alpha chain here, and human myoglobin. And see, these are the sequence of the amino acids here for our protein. Okay. Okay, and we're aligning them up so we can see if there's repeat patterns here. Okay? Which there are, by the way. Because, again, we have that same heme groups that are going to bind to oxygen, etc. So determining sequence identities. Sequence identities are going to be matched residues. As you can see, we actually had to frame shift these to actually figure that out. And so that's what you'd actually do is you take one, frame shift it with another, look for any matching. Okay. You don't normally do this by hand though, you do it with a computer. Yeah, a little easier now. So for the alpha hemoglobin and the myoglobin, your best alignment actually reveals 23 sequence identities, and that's these right here. So we take these, we slide past each other, and we can see we actually get two of them that are pretty good. We have 22 matches here, 23 matches here. This would be our best matching right there. And those are the results from a computer, by the way. So we have the number of matching versus your alignment. We'd have the computer, it would graph it, and we'd get these ones. Okay? And then we can look at them and go, oh, which one seems more likely? And then we have debates, and it becomes a fight. Well, you can insult me at a conference or something. Okay? Okay, anyways. I guess I'm done, right? In my time? Okay. Okay. So let's go ahead and call that a day, because we'll need to get over to the lab. And I need a little bit of time before lab starts.